Welcome to The Explainer. Have you ever thought about why you might have your dad's smile, but your mom's curly hair? How does nature actually mix and match all these traits? Well, today, we're diving into the really elegant rule that governs this whole genetic shuffle, the law of independent assortment.
And this really gets to the heart of the question, right? Are our traits inherited in these neat little packages? If you get your mother's eyes, is her hair color automatically part of the deal, or is it more like a random genetic lottery? This is the exact puzzle that Gregor Mendel wanted to solve with his pea plants.
So here's our game plan. We're going to start by tracking 2 traits, then we'll walk through Mendel's big experiment. After that, we'll decode the crazy pattern he found, understand the law it led to, and then, and this is my favorite part, we'll look at when that very rule breaks.
Okay, let's jump in. Section 1. Tracking 2 traits. See, to really unlock the next level of genetics, Mendel knew he had to stop looking at just one thing at a time, like plant height, and start juggling 2 traits at once.
So we already know Mendel had figured out the basics with his monohybrid crosses. He knew that for any single trait, you get that classic 3-to-1 ratio of dominant to recessive. But the really big question was still out there. What happens when you're watching 2 traits? Do they influence each other at all? And that very question leads us right into Section 2. Mendel's Dihybrid cross. This was the brilliant landmark experiment he designed to find the answer.
The setup is actually pretty simple, but totally brilliant. Mendel picked pea plants that were different in 2 very specific ways, the color of the seed and the texture of its skin. Focusing on these 2 traits at the same time was the key to everything.
He started out with what we call true breeding parents. So parent number 1 was what's called homozygous dominant for both traits, meaning it always made round yellow seeds. And the other parent was homozygous recessive, so it always made wrinkled green seeds. Now when he crossed these 2, the first generation, the F1s, they were exactly what you'd expect. Every single plant produced round yellow seeds. The dominant traits just took over. But, and this is the important part, genetically, these plants were now heterozygous. They were all secretly carrying the hidden instructions for wrinkled and green traits. The real test was, what was going to happen in the next generation? And this is where it gets really, really cool. Mendel then crossed these hybrid F1 plants with each other, and the results he got in this second generation were totally unexpected. It was like he'd uncovered a hidden mathematical code in nature.
Now, to understand what's about to happen, we have to think like a plant making its pollen or its eggs. The alleles for shape, the big R and little r, they have to separate. And at the exact same time, the alleles for color, the Ys, they also separate. And because of this, you can end up with 4 possible combinations in the sex cells.
So this grid here is a Punnett square. And I know it looks a little intimidating, but it's just a simple tool for mapping out every possible combination. Think of it like a multiplication table, but for genes. Across the top, you've got the 4 possible gene combos from one parent. Down the side, you've got the same 4 from the other parent. Each of those 16 boxes inside shows you what happens when a row and a column meet. This grid is really the key to seeing every single possibility. So after doing the cross and then patiently counting thousands and thousands of peas, Mendel found something amazing. The different types of seeds didn't show up randomly. Nope. They consistently appeared in this very specific ratio, 9 to 3 to 3 to 1. So the crucial point here is that this ratio splits into 4 distinct groups. You get 9 that look like the dominant parent, round and yellow. You get 1 that looked like the recessive grandparent, wrinkled and green. But here's the kicker. You also got 2 brand new combinations that didn't exist in the generations before, wrinkled and yellow and round and green. This was the proof that traits were not stuck together. This unbelievably consistent 9 to 3 to 3 to 1 ratio. This was the smoking gun Mendel was looking for. It pointed him directly to his second great law of inheritance, a real cornerstone of genetics that we still use today. And here it is. The law just says that the alleles for different genes, like seed shape and seed color, get sorted into the sex cells independently of one another. Basically, inheriting the allele for round seeds has absolutely no effect on which allele you get for seed color. They're independent. Honestly, this is the perfect analogy. Each gene is its own coin flip. The inheritance of seed shape is one coin, and the inheritance of seed color is a totally separate coin. That 9-3-3-1 ratio, it's just the statistical result you get when you have 2 independent coin flips happening at the same time.
But of course, in biology, discovering a rule is really just the beginning. The next fun step is to try and break it. And it turns out, the exceptions to independent assortment are just as important as the rule itself. You see, Mendel got a little bit lucky. The P traits he studied just happened to be on different chromosomes. But later on, research by people like Thomas Hunt Morgan on fruit flies showed that the 9:3:1 ratio didn't always work. And this forced scientists to ask a really critical question: what if the genes aren't separate coins? What if they are physically tied together on the same string?
And this idea is called gene linkage. It's exactly what it sounds like. When genes are sitting really close together on the same chromosome, they don't act like independent coin flips anymore. They tend to get inherited together as a block. And because of that, you end up seeing way more of the original parental combinations in the offspring than Mendel's ratio would ever predict.
Okay, so let's put this all together really quick. We saw how a dihybrid cross lets us track two traits, and how that reveals the elegant 9:3:1 ratio. We learned that this ratio is all thanks to the law of independent assortment, the idea that genes are inherited like separate coin flips. But we also saw that this rule has a major exception: gene linkage, which happens when genes are physically stuck together on the same chromosome.
And this leaves us with one last really amazing thought. Scientists like Alfred Sturtevant figured out that the frequency with which linked genes get separated is directly related to the physical distance between them on the chromosome. So the exception to Mendel's law didn't break it, it actually opened the door to a whole new field of science, mapping the precise location of every gene on a chromosome. How cool is that?